Dzień dobry wszystkim. Nazywam się Adrianna Niedzielska i wraz z Barbarą Dobosz mam przyjemność rozpocząć drugi dzień tygodnia Szkoły Doktorskiej SGGW. Wyłączony.
To jeszcze raz falstart. Dzień dobry wszystkim. Nazywam się Adrianna Niedzielska i wraz z Barbarą Dobosz mam przyjemność rozpocząć drugi dzień tygodnia Szkoły Doktorskiej SGGW, organizowanego w ramach projektu STER, Umiędzynarodowienie Szkół Doktorskich, finansowanego przez Narodową Agencję Wymiany Akademickiej. Będzie to dzień pełen fascynujących prezentacji i podróży naukowych przez świat badawczy naszej uczelni. Atmosferę zagęści konkurs, w którym przedstawiciele różnych dyscyplin naukowych zawalczą o miano najlepszej prezentacji. Zanim jednak zaczniemy, poświęćmy parę chwil na przypomnienie zasad. Każdy uczestnik mógł zgłosić do konkursu tylko jedną prezentację, której czas wygłaszania ograniczony jest do 12 minut. Każda dyscyplina oceniana jest oddzielnie, a prezentacja, by zostać nagrodzona, musi uzyskać minimum 15 punktów na 22. Oceniana jest forma prezentacji, estetyka pracy, wartość popularno-naukowa i warto zwrócić uwagę, że jest to kryterium, za które można uzyskać najwięcej punktów, dyskusja i czas prezentacji.
A teraz powitamy nasze szanowne gremium komisji oceniającej dzisiejsze wystąpienia, w skład w której wchodzą pani profesor, doktor habilitowana Małgorzata Kosicka-Kępska, Instytut Nauk Ożywienia Człowieka. Brawo! Doktor inżynier Kinga Noras, Instytut Rolnictwa. Brawo! Doktor inżynier Tomasz Raziński, Instytut Inżynierii Mechanicznej. Brawo! Oraz reprezentująca szkołę doktorską, doktor inżynier Agnieszka Otłowska. Brawo! I mamy również reprezentantkę doktorantów, doktorantkę magister inżynier Aleksandrę Jakimiuk. Brawo! W konkursie w naszych blokach startowych mamy następujących reprezentantów. Rozkład jest następujący. Nauki biologiczne 4 osoby, nauki rolnictwo i ogrodnictwo 3 osoby, natomiast równolegle mamy ekonomia i finanse oraz inżynieria lądowa, geodezja i transport po 2 reprezentantów, inżynieria środowiska, górnictwo i energetyka oraz technologia żywności i żywienie człowieka po 1 reprezentancie. I myślę, że na tym możemy zakończyć następ i przejść do pierwszego zawodnika. Pierwszym tutaj panem, który będzie prezentował, jest pan Maciej Piekarniak, reprezentujący nauki biologiczne. Brawo!
Dzień dobry. W dzisiejszej prezentacji chciałbym opowiedzieć o cyjanowodorze, który uznajemy obecnie za nowy środek, który może nitrować białka. Nazywam się Maciej Piekarniak, jestem z Katedry Fizjologii Roślin. Moim promotorem jest profesor Agnieszka Gniazdowska-Piekarska, natomiast promotorem pomocniczym profesor Urszula Krasuska. I na wstępie chciałbym opowiedzieć o cyjanowodorze. Cyjanowodór jest związkiem gazowym, który ma charakterystyczny zapach gorzkich migdałów i 2% roztwór wystarczy, żeby zabić dorosłego człowieka. Dodatkowo cyjanowodór łączy się z żelazem razem z cytochromem, oksydazą cytochromu C w mitochondrium, przez co inaktywuje oddychanie komórkowe. Cyjanowodór jest produkowany we wszystkich roślinach. Niektóre akumulują go w postaci glikozytów cyjanogennych i dodatkowo cyjanowodór powstaje na drodze Cyjanowodór powstaje także na drodze biosyntezy etylenu we wszystkich tkankach roślinnych. Jeżeli chodzi o największą koncentrację glikozytów cyjanogennych, możemy ją zaobserwować w bulwach kasawy, w migdałach, w bambusie czy w nasionach jabłoni. Cyjanowodór, oprócz tego, że jest uznawany za truciznę, może też pełnić funkcje regulacyjne, zwłaszcza u roślin i tutaj reguluje u roślin alternatywną oksydazę oraz szlak biosyntezy etylenu. Natomiast u roślin blokuje szlak syntezy ATP oraz transportu elektronów w mitochondriach. Wykazano w jednym z badań, że cyjanowodór może ulegać przekształceniu do innego ważnego gazotransmitera, którym jest tlenek azotu. I tutaj działa to na zasadzie hydrolizy katalitycznej lub katalitycznego utlenienia z wytworzeniem intermediatów, które następnie są utleniane do odpowiednich form tlenu, odpowiednich form azotu, tutaj tlenku azotu, dwutlenku azotu czy azotu atmosferycznego. Tlenek azotu uznawany jest za bardzo ważną cząsteczkę zarówno dla roślin, jak i za zwierząt. Jest to cząsteczka sygnalna, która pełni wiele funkcji w wielu procesach biologicznych i jest produkowana zarówno przez rośliny, jak i zwierzęta, czy bakterie i grzyby. U zwierząt jest produkowana przez syntazę tlenku azotu, natomiast u roślin mechanizm ten jest nie do końca poznany. I pierwszym doświadczeniem, którym chciałbym przedstawić, jest samoistne tworzenie jonów azotynowych, które powstają w wyniku zakwaszenia roztworu azotynu sodu i cyjanowodoru. I tutaj z zakwaszonych roztworów wydzielają się gazy, w tym przypadku donory tlenku azotu i cyjanowodór, które następnie w reakcji z wodą dejonizowaną mogą tworzyć jony azotynowe. Jony azotynowe następnie były oznaczane w wodzie dejonizowanej z użyciem odczynnika Grisa, mierząc przy odpowiedniej długości fali. I pierwsze wyniki, czyli tworzenie jonów azotynowych po traktowaniu gazami uwolnionymi z roztworu cyjanowodoru. Tutaj możemy zauważyć, że po godzinie inkubacji z tymi gazami w wodzie dejonizowanej wytworzyły się jony azotynowe po zastosowaniu 150 milimolowego i 300 milimolowego roztworu cyjanku. Natomiast po zastosowaniu 20, 50 i 100 milimolowego roztworu azotynu sodu również możemy zaobserwować samoistne tworzenie jonów azotynowych w wodzie dejonizowanej. Tlenek azotu jest jednym z ważnych czynników nitrujących białka. Może powodować nitrację tyrozyny, która jest uznawana za trwałą modyfikację potranslacyjną dla białek. Oprócz tego w procesie nitracji mogą brać udział też rodniki dwutlenku azotu czy nadtlenoazotynu. Czy nitrotyrozyna jest ważnym wskaźnikiem, często zwanym biomarkerem stresu nitrooksydacyjnego, który zaobserwowano w przypadku takich chorób jak reumatyzm, czy rak, czy choroby neurodegeneracyjne. I kolejnym eksperymentem, który wykonałem, żeby wykazać tworzenie 3-nitrotyrozyny z użyciem białka modelowego BSA. Tutaj przed właściwym eksperymentem dokonano redukcji mostków dzisiarczkowych z użyciem titioinu sodu i następnie BSA traktowano odpowiednimi roztworami zawierającymi cyjanek, tlenek azotu i była tutaj stworzona również kontrola. I po 30 i 60 minutach mierzono absorbancję. Czym tak właściwie jest BSA? Jest to białko wykorzystywane w wielu dziedzinach nauki, w biologii molekularnej, w biochemii. Składa się z 583 aminokwasów, gdzie 20 stanowią tyrozyny. I tutaj dobrze poznana struktura tego białka pozwala na opisanie mechanizmu nitracji. Jeżeli chodzi o wyniki uzyskane po traktowaniu BSA z odpowiednimi roztworami, dla roztworu cyjanowodoru można zaobserwować wzrastającą zawartość grup 3-nitrotyrozynowych w BSA po zastosowaniu roztworu cyjanku w stężeniu 70, 150 i 300 mmol. I ta sama sytuacja jest po zastosowaniu roztworu azotynu sodu. W tym przypadku roztwór był 100 milimolowy. Podobne wyniki, podobny trend zaobserwowano po wykonaniu analizy dot-blot, w której wykorzystuje się specyficzne przeciwciała anty-3-nitrotyrozynowe. I podsumowując, cyjanowodór można uznać za cząsteczkę, która powoduje nitrację białek, ponieważ wykazano, że podczas traktowania cyjanowodorem BSA powstaje grupa 3-nitrotyrozynowa. Dodatkowo pierwszy eksperyment pokazał, że po zakwaszeniu roztworu cyjanowodoru w wodzie dejonizowanej tworzyły się jony azotynowe, co wskazuje, że cyjanowodór może być źródłem jonów azotynowych w środowisku komórkowym. I właśnie prawdopodobnie cyjanowodór ulega przekształceniu do tlenku azotu w warunkach fizjologicznych w wyniku działania rodnika hydroksolowego lub anionorodnika ponadtlenkowego. Jakie to ma znaczenie dla przyszłych badań i dla rozwoju nauki? Przedstawione wyniki wskazują na nowy sposób, na nowy mechanizm działania cyjanowodoru na białka i można go później wykorzystać do zapewnienia bezpiecznych technik pobudzania nasion na szerszą skalę, co jest jednym z moich celów badawczych w pracy doktorskiej. Tutaj moja literatura i dziękuję za uwagę.
Dziękujemy bardzo. I do następnej prezentacji zapraszamy Ewę Szczucką, reprezentującą dyscyplinę Ekonomia i Finanse.
Jeszcze pytania, dyskusje jeszcze.
To przepraszam, to nie, to tutaj zagłębiamy się dalej w temat. I mamy chwilę na pytania.
Czy może Pan przytoczyć, jakie badania z zastosowaniem cyjanowodoru były już wykonywane i z powodzeniem zastosowane do pobudzania właśnie nasion? Czy są przykłady takich wyników badań?
Badania już były przeprowadzone przez jednego z naukowców, gdzie wykazał, że traktowanie cyjanowodorem, etylenem i tlenkiem azotu pobudzało nasiona kasawy do kiełkowania. I tutaj też wykazano, że cyjanowodór aktywował system antyoksydacyjny, przez co przyspieszał kiełkowanie i je dodatkowo polepszał. Więc to jest jedno z jedynych na razie badań przeprowadzonych, gdzie cyjanowodór został zastosowany jako jedna z cząsteczek pobudzających nasiona do kiełkowania.
A czy do nasion jabłoni były stosowane, do nasion jabłek?
Nasion jabłoni generalnie nie trzeba pobudzać. Tutaj nasiona jabłoni są moim materiałem badawczym, który stanowi modelową roślinę, żeby zbadać te mechanizmy. Natomiast do nasion jabłoni nie stosuje się tego sposobu pobudzania.
Dziękuję.
Czy ktoś ma jeszcze jakieś pytania może dla naszego prelegenta?
To może każdy pytać? Czy tylko
Tak,
komisja?
każdy.
To ja zapytam.
Proszę.
Chciałam zapytać o stężenie cyjanowodoru w na przykład Czy wiesz, ile takich migdałów trzeba by zjeść, żeby się zatruć?
Po pierwsze, migdały by musiały być już przejrzałe trochę, żeby ten cyjanowodór zaczął się po pierwsze z nich wydzielać i te glikozydy cyjanogenne tam były aktywne. Podejrzewam, że tak do 1 grama, nie znam dokładnej wartości, ale podejrzewam, że gdzieś tyle trzeba by było ich zjeść, żeby rzeczywiście się nim zatruć, ale to nie byłoby zatrucie, które by prowadziło do śmierci.
Aha, dobra. Dzięki.
My tutaj liczymy czas na telefonie. Nie pokazuje się czasu. No nie, my tu mamy kartkę 3 minuty, dlatego poszłam tam, żebyście widzieli, bo tu byście nie widzieli. No, dobrze, więc teraz w blokach startowych szykuje się pani Agnieszka Wal, reprezentująca nauki biologiczne, to zachęcimy może prawami, tak, na odwagę. O. Proszę bardzo.
So I would like talk about carnivorous plants, and my topic is digestion performed in pitcher fluid of Nepenthes. My name is Agnieszka Wal. I'm a third-year PhD student from Institute of Biology, and my supervisor is Professor Urszula Krasuska, and assistant supervisor is Dr. Paweł Staszek. Carnivorous plants can be found in swamps, wetlands, and tropical forest. Their habitat are poor in nutrients and the soil there is poorly aerated. As a result, carnivorous plants have limited root system. They use mainly the root system to absorb water and keep the plants in the soil. This environment forced the carnivorous plants to uncommon strategy of obtaining nutrients. They obtaining nutrients from the body of prey. As we can define the carnivore plant like an ability of plant to attract, kill, digest the prey, and absorb nutrients for the body of prey. 860 species of carnivorous plants are identified around the world. Among them, 169 species of pitcher plants, Nepenthes, are identified. But this number is not constant because all the time new species are discovered and some of them disappear from our planet. Nepenthes are carnivorous plants which have a trap at the end of the main nerve of the leaf blade. This is the passive trap filled with the digestive fluid. These plants have a passive trap, so they attract the prey by colorful appearance, sweet nectar, and smell. Nepenthes ventrata, which is the species I researched during my PhD study, is a natural hybrid of two species, Nepenthes ventricosa and Nepenthes alata. The species epithet is formed by combining two species name of parents, ventricosa and alata. Pitcher plants naturally occur in South Asia, in tropical forest, but Nepenthes ventrata and its parent species are endemics, and they can be only found in Philippine forest. But at our university in greenhouse, we have a great culture of Nepenthes ventrata, because we have more than 55 plants. They grow in pot with the acid peat, perlite, and sphagnum moss. The culture of Nepenthes ventrata is demanding because they are tropical forests, so they require a high temperature, around 28 Celsius degrees, and high humidity level, around 60%. This is the reason why the plants are hanged above the pool filled with the water. The trap of Nepenthes ventrata is filled with the digestive fluid. Digestive fluid consists mainly of water, but also enzymes. For example, proteolytic enzymes, phosphatase, and chitinase. Thanks to these enzymes, it's possible to digest the prey by the trap. In digestive fluid are also present low molecular weight compounds, for example, naphthoquinones. They have antibacterial and antifungal properties. Microorganisms such as bacteria from Enterobacteriaceae, Pseudomonaceae, and Yeserinaceae family are also found in the digestive fluid. Some mineral compounds like magnesium, sodium, and potassium are also present in the fluid. Digestive fluid have unique physicochemical properties, like lower surface tension than water and high viscoelasticity. Thanks to these properties, it's challenging for the prey to escape from the trap, and the plant achieves her goal and can digest the trap and obtain the nutrients. The experimental model I used to measure the chitinolytic and proteolytic activity is the developmental model, which means that digestive fluid was collected from the trap at the different stage of development. The plants are randomly selected to the experiments, and one of trap from each plant was tested. Digestive fluid was collected from the closed trap, a trap which develop open but not fed, trap develop opened and fed with the aqueous solution, which is the source of protein, and the trap with the signs of aging, for example, dry lid. During this experiment, the opening of the trap was covered with glaze to prevent the falling prey into the digestive fluid. Protease, the enzymes that break down protein into polypeptides. This reaction is stimulating by water. So I measured the proteolytic activity in the digestive fluid of Nepenthes. And after the trap is open, the proteolytic activity is higher in this trap. So the reason of this can be related with the ready of this trap to digest the prey, or that the trap is in this moment during the digestive process. Enzymes inhibitors is a molecule that binds to enzymes and block this activity. And enzymes can be specified to some selective products, so they block a specific reaction. For example, protease inhibitor limited the proteolytic activity. And after application, the inhibitor of proteolytic activity proteolytic activity was measurement in the digestive fluid of Nepenthes, and only decrease of this activity is shows in the trap opened and developed. This result shows that the proteolytic activity in the digestive fluid of Nepenthes is not only related with the enzymes secreted by the plants, but also with the microorganism present in the digestive fluid. Chitin is a polysaccharide, and this is the main component of the exoskeleton of insects and cell walls of fungi. Chitinase are enzymes which breaks down glycoside bonds in chitin. Nepenthes secrete into digestive fluid chitinase, and this chitinase break down the chitin into chitin oligosaccharides. I measured the chitinase activity in the digestive fluid of Nepenthes, and I found out that the level of activity chitinase was high in the digestive fluid from developed trap, and only from the trap with the signs of aging. The high level of chitinase activity in the trap with the signs of aging can be related with the protect and antifungal activity of these enzymes, because in the digestive fluid of the trap with the signs of age can be a great habitat for the fungi, but chitinase can break down the chitin in the cell wall of this pathogen. In digestive fluid of Nepenthes are chitinases secreted by the plants, but also they are bacteria which characterized by the chitinolytic activity. So the chitin is breaks down by chitinase into chitin oligosaccharides, and bacteria breaks down the chitin oligosaccharides to glucosamine monomers. Thanks to the presence of the bacteria and plant enzymes in the Nepenthes digestive fluid, it's not necessary that plants secrete all enzymes to digest the prey completely and absorb nutrients. Thanks to the presence of bacteria and enzymes, the process of digestion have place, and the plants can obtain the nutrients and achieve the success and grow. In addition, thanks to the antifungous properties of the enzymes protect the plants from the pathogen, and the plants grow healthy and beautiful. Summary the results: the application of the protein increase the protease activity. Plant protease inhibit slightly reduce the activity of proteolytic enzymes in the digestive fluid, and the presence of enzymes and bacteria in the Nepenthes fluid allows to the food completely digestive, absorb the nutrients, and protects the trap and digest fluid from the pathogen. Thank you for your attention.
So, some question? Okay. Podacie tam?
I would like to know, are carnivore plants beneficial for environment and safe for human?
They are beneficial for environment because they reduce the population of insects. So it's quite popular to use carnivorous plants in some botanic garden, but they are also very beautiful. So they are nice to admire the appearance and show, but they are safe for human because the enzymes they produce is in the solution of water, so it's not a higher concentration of these enzymes, and these enzymes can be used, for example, in some cosmetic maybe in the future, or maybe to proteolytic a little some food, for example, some fish in the process of production or something like that.
Oh, maybe I would like ask the question: how large the tested sample was? How many of plants you get to receive this fluid?
I take the fluid from one trap, but I do this in 5 or 6 biological replication. It depends on the necessary and statistic evaluation.
Okay, thank you.
Maybe something connected to this the first question. Are humans already using those plants for something?
Yes, people use the carnivorous plants in traditional medicine, and it's possible to isolate some enzymes. But nowadays, still we have some research about the use carnivorous plants and enzymes in antibacterial properties and anti-cancer properties. But this research are only in the laboratory nowadays.
Okay, thank you.
Okay, so if we have any questions, so the next, Ada?
Jak widać, bardzo nie mogłam się doczekać tego momentu. Do następnej prezentacji zapraszamy Ewę Szczucką, reprezentującą dyscyplinę Ekonomia i Finanse, która przybliży nam temat jeszcze nie tak popularny w Polsce i zaprosi do poznania tajemnic warmińsko-mazurskiego ośrodka oferującego terapię z udziałem krów.
Good morning, everyone. My name is Ewa Szczucka, and today I would like to present you some selected results from my latest research, which I conducted in March and April this year. The study is about the determinants of cow-assisted therapy provision in Poland. At the beginning of my talk, I will say about what cow-assisted therapy is. Then I will outline the aims of the study and the method which I used. After that, I will discuss the studied object and the results of the research. And finally, I will briefly summarize my presentation. We should know that the therapeutic properties of domestic cattle were already known in ancient times, but cow-assisted therapy in its current form, in its current shape, was born in the Netherlands around 2010. Since then, cow-assisted therapy has become popular in many countries, such as USA and Switzerland. And other names for cow-assisted therapy are cow-based therapy. It can be also named cow-cuddling therapy or cow-hugging therapy. The etymology of the term derives from two words. First is the cow, which is the adult female of domestic cattle. And second word is the Greek word therapia, which means therapy or treatment. But cow-assisted therapy can be carried out not only with cows, but also with heifers and calves. So, in short, cow-assisted therapy is a process of health support in which a representative of domestic cattle is a central and indispensable element. Now I will talk about some of the benefits of contact with cow. Cow's heart rate is lower than humans. They also have higher temperature. So when we cuddle with cows, it helps us to relax and also distress and slow down and calm down. We can also mention about talk therapy aspects because people can talk to cows, share with them their problems, and animals don't judge. Also, the place in which the therapy session takes place is very important because if it takes place in a meadow among greenery and singing birds, this also increases the therapeutic aspects. In addition to this, cow cuddling can be used to help treat people with various disorders and diseases. It can be helpful in the treatment of sensory integration disorders, for example. Now we can move on to the next part of my presentation. The main objectives of my research were to identify the determinants of cow-based therapy provision and to specify the barriers to the development of cow-cuddling therapy in Poland. The method which I used is case study, and it included one structured interview and 3 in-depth interviews with service providers. I also interviewed one visitor, and despite I used participatory observation and analyzed service providers' social media site. The establishment selected for the study is an organic farm located in the village of Sterławki Wielkie in the Warmińsko-Mazurskie Voivodeship. The owners are a married couple. It's Mrs. Agnieszka and Mr. Adam Scibut. And currently it is one of few in Poland and the only facility in the region providing this type of animal therapy. And also this is the place where heifers trained for therapy purpose can be purchased. The therapy is conducted with white-backed cows and calves. This is indigenous Polish breed, native Polish breed, and its name white-backed comes from the characteristic coloring on its back, as we can see on the first photo. Animals at the facility are kept in free-range system, and this is very, very beneficial for their physical health and well-being. During therapeutic activities, visitors can stroke the cows, cuddle with them, and sit next to them in the middle. And in the future, service providers plan to expand the activity elements to include cleaning the animals, learning to lead them on a halter, and counting dots. There are also plans to introduce support equipment such as sensory balls. Cows used for therapeutic purpose should have the right characteristics. First of all, they must not be skittish or aggressive. Instead, they should be friendly, they should be calm and enjoy the company of people. And also, they must be well trained. When we are speaking about financial aspects, one adult cow generates for the object cost of little bit more than 900 Polish zlotys per year, and it includes feed, supplements and mineral leaks, and veterinarian services. Mrs. C would know how to correct hooves by herself, so expenses related to animal care are reduced. And now revenues. The price for 1 hour of individual classes is 50 zloty per person, and for group classes, which can be attended by up to 4 people, the price is 40 zloty per person. For some cows, the service providers get a subsidy 1 a year under the Conservation of Animal Genetic Resources program. I also identified barriers to the provision of cow therapy, and here are listed 4, in my opinion, the most important. The first is the cultural barrier. That results in the low popularity of this form of therapy in Poland, because Polish people have the belief that domestic cattle are bred only for meat and milk. And service providers here in the facility have often been met with astonishment and shock by people when they tell them about their therapeutic cows. Other barriers are related to finance. On the one hand, there is the economic hardship of society, which may translate into low demand for therapeutic services. And on the other hand, the owners of the facility don't currently have spare funds that they could use to develop their health service. And now personnel barriers are related to the difficulty of finding a trusted person with the right qualifications to assist in therapy session. And the last mentioned barrier here, the climate barrier, which is important because of free range rearing system and low temperatures in Poland. And now we can come to the last part of my presentation, the conclusion. We should remember that cow-assisted therapy has many benefits for health, for our well-being. White-backed cattle is very suitable for therapeutic purpose, but the most important issue is proper training of the animal. Despite growing popularity of cow cuddling around the world, it remains relatively unknown in Poland. And this is due to the barriers that exist in Poland. And here are 2 slides with the literature and other sources. And here's the QR code to the provider's Facebook page. If anyone is interested, you can scan. And thank you very much for your attention. Please feel free to ask me questions, maybe in Polish, I prefer. Thank you very much.
Bardzo ciekawy temat. Ja nie wiedziałam, że jest taka forma terapii z krowami, a w Polsce jest zainteresowanie na to, tak jakby, wiesz?
Ja od 2 lat staram się, tak, dziękuję bardzo, to jest dobre pytanie. Od 2 lat staram się namierzyć takie miejsca, które świadczą w Polsce krowią terapię. I jak na razie właśnie namierzyłam ten obiekt, do którego pojechałam i tam zrobiłam badania. Innym miejscem, a tak właściwie krową terapeutką jest krowa Mania, to jest krowa w Fundacji Bękowo pod Poznaniem i ta krowa przez wiele lat jeździła do przedszkoli, do dzieci tam, żeby dzieci mogły mieć z nią zajęcia. Teraz już stacjonarnie tam przyjmuje gości. I tak więcej jakby miejsc na pewno są, ale nie znalazłam, mimo że szukam poprzez mapy Google, poprzez serwisy, poprzez Facebooka i nie znalazłam. Więc myślę, że to jeszcze jest duży potencjał tutaj dla Polski, właśnie potencjał dla jakiegoś może regionu, gospodarstwa, które by chciało wprowadzić taką krowią terapię, żeby zainteresować osoby, bo naprawdę temat jest mało popularny w Polsce i budzi duże emocje.
Dzięki wielkie.
Którą barierę uważa Pani za taką najważniejszą w tej krowiej terapii? Czy to są raczej te koszty ekonomiczne, czy może strach przed tak dużym zwierzęciem, czy może jeszcze jakaś jest inna bariera, która jest taka istotna w rozwoju tej krowiej terapii, że tak się wyrażę?
Jak dla mnie to jednak te bariery kulturowe, czyli że my tutaj w Polsce nie zdajemy sobie sprawy, że my to bydło domowe, te nasze krowy możemy również używać do celów terapeutycznych, żeby polepszyć swój nastrój, swoje zdrowie. I nawet dlatego tego po prostu nie szukamy, tak? Też, tak jak wspomniałam, tutaj właściciele obiektu często, jeżeli mówią jakimś osobom, znajomym, że mają takie krowy terapeutyczne, że zapraszają na terapię, to często to spotyka się z takim nawet niedowierzaniem, tak? Ale że jak to, że krowa, że na terapię, że przecież krowa to jest na mleko, na mięso, a nie żeby to było zwierzę, dzięki któremu my możemy się poczuć lepiej, tak? Więc tutaj jakby te nastawienie nasze w Polsce myślę, że jest kluczowe, bo w innych krajach krowia terapia odniosła sukces. Na przykład właśnie w Holandii, gdzie się narodziła, w Szwajcarii czy w Stanach Zjednoczonych są takie obiekty. Więc myślę, że te kulturowe bariery, jeżeli chodzi o taki strach przed zwierzęciem, to rzeczywiście, jak tutaj się pytałam pani Agnieszki, czy odwiedzający odczuwali jakiś lęk, to tak, zdarzało się, ale szybko przezwyciężali, tak? Podczas jednej sesji już osoby, dzieci, czy nawet dorośli, którzy bali się podejść do takiej krowy, już potem podchodzili, bawili się z nią, głaskali i przytulali.
Chciałbym się spytać, czy to są jakieś specjalne rasy tych krów? Czy generalnie na jakiej podstawie one są dobierane? No bo jednak to jest dosyć duże zwierzę, one może być niebezpieczne nawet, nie? I dlatego chciałem się o to spytać.
Tak, to też jest bardzo ciekawe pytanie. Ja tutaj jak szukam obiektów po całym świecie, tak, przeglądam serwisy, strony internetowe, które właśnie taką terapię oferują, to rasy są naprawdę różne krów. Tutaj polska rasa rodzima białogrzbieta okazało się, że bardzo się fajnie nadała do tych celów terapeutycznych, jednak to myślę, że głównie ze względu tego, że te krowy, te zwierzęta tutaj były ze swoimi właścicielami właściwie od cielaka i pani Agnieszka włożyła wiele pracy, żeby przyzwyczaić je, żeby odczulić je na dotyk, żeby nauczyć je chodzenia na kantarze, przytulania, tak? Czyli ja tutaj myślę, że może nie konkretna rasa jest ważna, a to, żeby to zwierzę jak najlepiej przygotować i już od małego cielaka.
Ja mam takie pytanie, czy szukając tych ośrodków terapeutycznych z krowami, spotkałaś się może na inne zwierzęta gospodarskie wykorzystywane właśnie w terapii? Myślę tutaj sobie troszeczkę o kozach, nad którymi sami tutaj trochę debatujemy w kontekście nawet Dni SGGW, bo zapraszamy Państwa na jogę z kozami w piątek i w sobotę. I ja gdzieś tam troszkę to śledzę, natomiast może Ty tutaj naukowo bardziej byś nam coś powiedziała na ten temat. Wiemy coś o wykorzystaniu kóz lub innych zwierząt gospodarskich?
Tak, tutaj kozy jakby się wpisują też szerzej w ujęcie bowiterapii, czyli to będą kozy, owce i krowy właśnie. Typowo kóz w Polsce akurat nie szukałam, ale szukając krowiej terapii natknęłam się na bardzo fajny obiekt w Stanach, Goat Yoga Arizona, i to właśnie tam oni szczególnie prowadzą goat jogę, a oprócz tego mają krowią terapeutkę Maisie i tam właśnie się dowiedziałam, że kozy oprócz tego, że możemy po prostu je obserwować, głaskać, tak mieć z nimi kontakt, to jeszcze mogą tutaj nam posłużyć jako asystenci w naszej jodze, tak? Tam nawet było na filmie, który oglądałam od Goat Yoga Arizona, że te małe kózki skakały ludziom po plecach, więc to też było takie fajne, rozkoszne. A w Polsce, jeżeli chodzi o terapię z udziałem kóz, nie natknęłam się jeszcze na taki ośrodek, że tak typowo jakby określa, że to jest terapia i że zapraszają do swoich kóz.
Bo badania naukowe pokazują, że rzeczywiście obcowanie z tym zwierzęciem, z kozami, przynosi pozytywne aspekty dla naszego zdrowia psychicznego, więc może jest to jakaś ścieżka drogi dla naszej uczelni.
Dobrze, to jak zakończyliśmy już dyskusję, to gratuluję tutaj naszej prelegentce, tak, wielkie brawa. No. I kolejną tutaj osobą będzie reprezentantka inżynierii lądowej, geodezji i transportu, magister inżynier Luiza Rzepczyńska. Brawo! Tak. Proszę bardzo.
Dzień dobry. So I will also try to present in English, just to try myself. I would like to tell you today a little bit about the physical and mechanical properties of cohesive soil geopolymerized with the fly ash. Here a short table of contents, and I think we can all agree that we live in a time when we are not really thinking where to build, but instead, we're just trying to build everywhere, which is somehow sad. But the fact that we're trying to build everywhere means that we have to somehow make the grounds that we are trying to build to improved so that stable engineering structures can be made. Different types of ground cause different type of problems in the engineering field, and I'm working with so-called expansive soils, which are those that change its volume with the addition of water. They may cause, because of that, numerous geotechnical problems and made damaged foundations subjected to that extensive deformations because of addition of water. And that may result in cracks. To sustain those volume changes, we are trying to apply some sort of stabilization, or we are trying to even replace the whole ground that we are building on. And those are quite expensive. So the chemical stabilization of those problematic soils not only can reduce those volume changes that are very much not wanted, but also improves the engineering properties such as mechanical and physical of those improve. One of the most common and widely used stabilization is the one with cement and lime. It's effective and fast, so it's widely used. However, both of those very negatively affect the environment due to the high carbon dioxide emissions during their productions. It is actually estimated that even 5% of the whole human-made CO2 emission is because of the cement production. So that's why the alternative approaches to stabilize the expansive soils are being developed. One of the particular interests are so-called industrial solid waste, such as fly ash, for example. That is an aluminosilicate-based material, and it's mostly used as the addition to the concrete. However, fly ash in the presence of sodium hydroxide, so very alkaline environment, can break those silicon aluminum ions, which leads to the creation of so-called inorganic binder geopolymer. Geopolymerization is a process reaction of aluminosilicates and oxides in that very alkaline environment, and the geopolymers itself are 3D polymeric chains that are made because of the dissolution of those silicon and aluminum oxides bonds. Then the gel is formed, and with the time, polymerization happens. So what I did in the topic I would like to present for you today is I mixed the following materials, that is bentonite, kaolin, fly ash, and sodium hydroxide. The mixed proportion of the samples I tested are in the table below, and all of those components were carefully mixed in the designated proportions in the air dry conditioned state. To form those samples, I compacted them in a cylinder using the Proctor hammer with the standard energy of compaction, that is 0.59 joules per cubic centimeter. Oh, and those samples were cured for 7 and 14 days. But before those, I needed to determine the granulometric composition of the dry non-geopolymerized mixtures, also check liquidity and plasticity limit. And not so fun fact about this test is that it's standardized, but it basically, we have to form a little ball of the sample, and then as we can see those rolls, and we're repeating those to check when the cracks started to happen. So it tells us when the ground is getting from the hard plasticity to semi-solid state. Also, the free swelling in Vasilev apparatus of the geopolymerized and non-geopolymerized soils, as well as the uniaxial compressive strength, like I said, on the samples that were created in a cylinder using a Proctor hammer with the energy of the geopolymerized and non-geopolymerized samples. And the results, we see the granulometric curves of the soils mixture tested, and for a non-specialist it may look pretty similar. However, only the mixture with non-addition of the fly ash and with the addition of 8% of the fly ash were decided that they are silty clay, and the ones with the addition of 16% and 24% of the fly ash were decided that they are clayey silt. This diagram shows free swelling of the soil mixtures, and we can see that both of the axes are in the logarithmic scale. So those differences here are actually quite a lot, and none of the cured samples swelled by even 0.5 millimeter over 2 days that the test took place, which is compared to the initial height of the samples in 20 millimeters, meaning that the swelling was less than 2.5%. We can say that it's almost gone. For comparison, a sample that was natural soil, contained only kaolin and bentonite, swelled more than 8 millimeters, which is over 40% in those 2 days. Here in the table 2, we see the results of the Atterberg limits of those soil mixtures, which is plasticity limit and the liquid limit. we can see that the plasticity limit decreases with the addition of fly ash to the base mixture. The only exception is that second sample with 8% addition of the fly ash. However, as I said, that test, however standardized, it may not be very accurate. However, the liquidity limit tests of those individual mixtures, we can see that the liquidity limit decreases on average by 20% with an increase in the amount of fly ash added. Every step was 8 percentage points. Here we can see those liquidity limit results on the graph. And considering the decrease in both liquidity and plasticity limit, the most important conclusion is that reduced soil plasticity improves its workability, so the creation of the samples is much easier. Here we see the compressed strate of non-geopolymerized and geopolymerized soil mixtures. I also wanted to focus your attention on the axis, on the x-axis. As we can see, those data is obtained as much higher. And also the graphs of the plots of Young modulus of those non-geopolymerized and geopolymerized mixtures. Also want to focus your attention on the obtained results, that it's 10 times higher than of the cured mixtures than the non-cured mixtures, and the best strength and stiffness results were shown by geopolymerized mixtures that were having the largest amount of reactive aluminosilicate substances, which means that the most of the fly ash was added, and those were compacted at the moisture levels closest to their plasticity limits. Yes, so the mixtures 3 with the 16% addition of the fly ash and 4 with the 24 addition of the fly ash were compacted using the least amount of water, and after 2-week curing period showed compressive strength ranging from the 127 to 144 megapascals. However, it's important to notice that those were cured for 14 days, not even as the standard would assume 28. And with the geopolymerization, it's that they're improving their mechanical and physical properties even a year after making of those samples. And that those results corresponds to the strength class of C1.2, 1.5 cement stabilized soils. In the conclusions, the soil swelling decreased by the 95 to even 99% in case of the geopolymerized soil after 14 days of treatment, which from the practical point of view is a complete reduction of the swelling phenomenon, and the mechanical properties in the form of uniaxial compressive strength increased from 0.22 megapascals from the natural soil non-geopolymerized to 1.45 megapascals for the one that was stabilized with the geopolymerization. And that standard written there introduces the possibility of using hydraulic road binder for soil stabilization. And the tested geopolymerized mixtures, according to these standards, could theoretically be applied to improve substrates for roads KR1 to KR7, KR meaning category of the movement, basically. And overall, I think that geopolymerization is a viable alternative to that classic chemical stabilization methods, and will therefore increase the land available for construction, reduce the risk of damage to the buildings and roads, and also decrease the CO2 emissions to the atmosphere. Here are references, and thank you.
So now we can go to discussion. So maybe somebody have some question?
I have just one favor. I also would prefer the questions to be in Polish. Thanks.
Co my jako istoty ludzkie powinniśmy zrobić, żeby oszczędzać tą ziemię przed degeneracją, przed niszczeniem? Czy są jakieś takie Pani najważniejsze wskazówki, obserwacje? Co mówi na ten temat literatura?
Wydaje mi się, że to pytanie jest nieco niepowiązane z moim tematem, ze względu na to, że mogę sama sobie podstawić, przepraszam bardzo, nogę, ponieważ moim zdaniem im więcej tej naturalnej przestrzeni, tej przestrzeni z drzewami, z krzewami, która będzie tak naprawdę najbardziej naturalna i co prawda być może mniej w obecnym świecie pożądana ze względu na to, jak przemysł się szybko rozwija i jak wszędzie się śpieszymy, jak wszędzie musimy szybko dotrzeć, więc tych dróg nam jest potrzebne bardzo wiele. No, moim zdaniem komunikacja miejska jest naprawdę na tyle dobrze rozwinięta, przynajmniej w Warszawie, że tak trochę dywagując, że naprawdę świetnym pomysłem byłoby ograniczenie ruchu drogowego.
Czy wiadomo coś na temat CO2 w przypadku zastosowania tej mieszanki? Na ile ekologiczniejsze, czy tym lepsze pod kątem właśnie...
Tak jak mówiłam, te 5% ogólnej emisji CO2 wywołanej przez człowieka jest związana z produkcją cementu. W przeciwieństwie do również wydobycia materiałów, które są potrzebne do produkcji betonu typowego z cementem, ten materiał tak naprawdę powstaje po spalaniu węgla, więc on jest produktem ubocznym tego, co i tak już się dzieje, przemysłu energetycznego. To jest odpad, który używając znajdujemy dla niego ponowne zastosowanie i go recyklujemy.
Okay.
Bodajże tuż przed wnioskami był taki slajd z modułami Younga. Tam pierwsze, co zwróciło uwagę moją, to po prostu one są takie dosyć małe, te współczynniki, bardzo małe. I z czego to wynika? Może coś?
Z czego wynika? Wynika to z tego, że to nie jest materiał, który będziemy używać do budowy samych dróg. Jest to materiał, którym stabilizuje się podbudowę pod te drogi. I grunt, na których dopiero te kolejne warstwy drogowe są położone.
To był moduł Younga niezagęszczonego materiału? Nie bardzo.
Zagęszczonego.
Dobra.
One nie są faktycznie bardzo wysokie, natomiast zależało mi na tym, żeby pokazać różnicę między gruntem pozostawionym samym sobie, również zagęszczonym, natomiast nie nieustabilizowanym w żaden sposób, a tym poddanym stabilizacji.
Dobrze, dziękuję.
W takim razie bardzo dziękujemy. Proszę o brawa. I by zagłębić się w tajniki dyscypliny inżynieria środowiska, górnictwo i energetyka, zapraszamy do wysłuchania prezentacji Poja Gezel-Aja. Temat prezentacji jest odpowiedzią na zmiany klimatyczne, z którymi mierzą się takie ekosystemy jak torfowiska. Zapraszam.
Hello everyone, my name is Puya Gezel-Aja, second year PhD student of environmental engineering, mining and energy. Since we are in different fields, I decided to present my presentation in a different way. First of all, I need to introduce the importance of peatlands for you, and so that you can realize that how much is it important for research. So I would like to draw your attention to a reel. I made it for this contest as well for this purpose.
Peatlands store large amounts of carbon.
We don't have
Peatlands store large amounts of carbon. The largest terrestrial natural carbon storage in the Earth.
But it has something.
They provide essential habitats for a wide range of plant and animal species, supporting biodiversity. Peatlands also play a crucial role in water regulation, helping to prevent floods and maintain water quality. Cultivation activities on the one hand, and global warming on the other hand, have led to the degrading of these natural lands. Consequences of peatland degradation. Emission of the carbon locked within them to the atmosphere, and speeding up the climate change. Biodiversity loss. Flood protection loss. We must take immediate steps to protect and restore peatlands. Let's work together to ensure a sustainable future for peatlands and our planet. Peatland.
I hope now you have been convinced. It is very important and very essential ecosystems. They are peatlands, and we have to take immediate conduct and research on them. But the problem is conducting research in peatlands are very difficult. You wanna know what I am talking about? Let's just take a look at it.
Trousers are getting dirty. Look at the state of my feet. I've got to try and get over there.
It's not funny at all, especially in the field and in the cold weather. I'm telling you, it's very hard to conduct research in the peatlands. Also, if you can survive and you can enjoy your fly back to your home and take a lot of proud of yourself and make a lot of Facebook posts and something. But this kind of difficulty is not the only ones. There are in other reasons, like time consuming, cost consuming, unreachable places, and unreachable places in the field research, especially in the large areas. That makes me and my supervisor to find an alternative way for conducting research in the peatlands. These days, the remote sensing techniques have revolutionized our understanding of any aspect of the environment, and we decided to use, and the question was that how can we use the remote sensing in monitoring peatlands health? That was our question, and this question is rely on two hypotheses, which is a remote sensing technique such as INSAR techniques can be used as estimating for peatland subsidence, and peatland subsidence can be used as a proxy for peatland health. We choose the Biebrzα National Park, Biebrzα Valley, as our case study. It located in the northeast of the Poland, and as you can see in this case study, we have access to observation data, which are using with this piezometer, and it is the mechanism of the piezometer, and that is the distribution of the piezometer in the or case study. I'm not going to go to technical detail of our framework. We want to actually we proposed a framework for doing this kind of research, and I just want to give you a very briefly summary about that framework. It's based on the Alaska Satellite Facility, cloud computing in SAR on demand. It's very sophisticated name, and data, and seasonal annual search approach and optimization process for estimating the subsidence of the area in the large scale. By implementing our framework in Biebrzα National Park, Biebrzα Valley, we revealed a tragedy in there. We revealed that the peatland subsidence rate of almost 1.5 centimeter per year, which means in average 46 tons per hectare are disappearing in these lands. And we also, by using that piezometers, verified our approach or framework. As you can see, it is a very reliable result. In conclusion, I would like to close my presentation with two news: bad news and good news. The bad news is, despite the fact of high conservation in Biebrzα National Park, Biebrzα Valley, peatlands are not in a good condition. They are, in general, in a poor condition. And it means they emitting carbon dioxide, that biodiversity is on the dangerous, and flood protection is going to last. It's not only for disappearing of these lands. And it deserves 1 minute silence for this, but we don't have time for it. So I want to also go for the good news, which is about a publication of this research in scientific paper is in Ecological Indicator journal, for which I need a couple of days to do that. Thank you very much.
Zapraszamy zatem tradycyjnie do pytań.
If I may, I have I have a quick question. I know that these results are based on the Biebrza Valley, but do you have any thoughts about peatlands in general? Maybe not specifically in Poland, but around the world. Are they also subsidized, like you've told us in this case?
Yes. We work on peatlands in Norway, and that picture you saw, it was in Norway, and yes, there are a lot of sites we are working dealing with, especially in Norway and Netherlands, and we conducting research in there. And the good thing about this approach is that you can use it in all of the world, and it doesn't matter where you are.
Can something be done for the peatlands to be improved? You told us that they are getting lower and lower. Can something be done?
Yes, the main reason of the peatland degradation is on two factors. One of them is the agriculture activities, and another one which we can protect the lands with some policy and something like this. But another reason which is very important is related to climate change, and it's inevitable, to if we want to protect these lands, we want to have a big picture and doing some effort in the emitting for climate change.
Maybe it works. How? And Puya, how you assess the condition of the peatlands? Because maybe I didn't get this information from the presentation. How you?
I bring them for you. As you can see, we have some blue point and some orange point. The blue point is, first of all, I need to tell that it's the data of one field of research. That's distributed points. I show you in, let's see, these piezometers we used for changing the elevation of the soil, and as you can see, the blue points are the trend of observed data, and the trend of the orange one is the obtained from satellite imagery, and we test and verify in these 11 points, and we find that it's very reliable. As you can see, the RMSE of 2 millimeter is very, very good and reliable. Dziękuję.
Dziękujemy bardzo, wielkie brawa. Po tak głębokim skoku w teren myślę, że przyda nam się chwila oddechu, dlatego zapraszam Was na 10-minutową przerwę. Witam wszystkich po przerwie i przechodząc do tematu rolnictwa i ogrodnictwa, swój temat zaprezentuje Abu Zargafur, rzucając światło na złożone interakcje w uprawie zbóż. Serdecznie zapraszam.
Dzień dobry. Thank you very much. My name is Abu Zargafur, as she mentioned. I am from Department of Biometry of Institute of Agriculture. So I'm going to present the influence of crop management on stability, sickle, cereal yield, and grain quality traits. These are the following 5 contents of my presentation. So start from the introduction. I'm working on the rye crop. It's very important because in many countries, it's a very rich source of fiber, phytochemicals, and bioactive compounds, which are very important for our health point of view. So yield and quality are very important when we consider its cultivations. So their higher yield and quality are desirable because they allow the farmers to maximize their profits by producing more grains with the minimum utilizing the resources. So, but their quality and yield are affecting due to the changes in the climatic events. So this picture is showing some climatic events. You can see there are increasing number of extreme climate related events in upcoming years. So I think from 1990 to 2016, which are increasing. So these changes would severely affect the yield and quality and stability of many kinds of field crops. So recently, European agriculture sector faced very pronounced climatic changes than the long-term average in a few decades. You can see in these pictures, this is a warm spell duration index, and it is predicted that in upcoming decades it will increase. So it's a very severe condition. So because every crop have special climatic related indicators to produce more yield and quality, so we need to focus on and to consider. About 47% of European area have received less rainfall than usual, and about 17% of the vegetation is negatively affected due to this climatic and especially due to the drought. And in this picture, you can see the red areas which are exposed to these drought conditions. And many countries of the Europe are affecting due to this drought. So when these plants are subjected to these combined multiple stresses, their metabolic and molecular pathways are disturbed. So, and because these crops are required special climatic events to produce more and higher quality products. So whenever we consider this, so it's very important to find genotypic adaptations for addressing the future climatic changes. So it's very important to produce climatic resilient and stable genotypes which have higher yield and quality, and also very important to ensure the food security in the future to tackle these environmental changes. So second part is our objectives. So my first objective is to evaluate the grain quality traits and stability of winter rye by using the different kind of statistical parameters, and also to assess their relationship between the grain yield quality and cultivar characteristics, and to compare the contribution of yield components to the grain yield variation between the population and hybrid cultivars. So let's move to the third, what we use in our experiment. Yeah, our experiment is 2 factorial. It's 2-year data that we obtained on the 3 locations of Poland. And we use 2 management levels. One is moderate input management, and the second is high input management. In the moderate, we use 90 kg nitrogen per hectares, and in the high input, we increase the 30 to 40 kg nitrogen per hectares. And you can see in the left corner of the cultivars, there we use 15 cultivars, which and some are hybrid and some are population. So these are the agronomic and quality traits which we determine by using the near infrared spectroscopy method and also using the classical approach. So the analysis of the grain yield and its components and also the grain quality traits were performed using a single stage approach. We use a linear mix model. And also we assess their stability by using the Shukla stability variance and the multi-trait stability index to evaluate their stability. And moreover, we use the classification and regression tree methods to assess their relationship between their stability and their characteristics. So all of these kind of analysis were performed using the R software. So this is the equation of linear mixed model which we use in our experiment. So let's move to the results. Yeah, these are the results. There are two kind of tables. One upper is the moderate input crop management, and lower table is the high input crop management. So the red boxes are showing the mean higher values of these traits. And you can clearly see upper and lower table, different cultivars give us the different higher mean values of these parameters of agronomic and quality traits. So this is the path analysis diagram, which also shows us the influence of grain yields and its components and the relationship between them. So you can see in the red circle across the management and cultivars, which the number of spikes have a higher influential value, which influencing the yield, and the later than is 1,000 grain weight and the number of grain per spikes. So this is also a comparison of cultivars order of the value of ranking Shukla stability variance and in both crop managements and the types of the cultivars. And you can see in the first moderate input crop management, which is MIM, scanned cultivar which is in a box, is ranked second, but in a high input crop management is ranked last. So you can clearly see the difference of these management effects on the performance of these cultivars. And this is the regression tree CRT method, which we use to to evaluate the sum of the ranking of the Shukla stability variance and multi-trait stability index of winter rye cultivars. In the first line, you can see there are boxes. In the box, the upper value is showing the mean value of the stability parameter, and the lower value, which is in the percentage, it's represent the percent of the cultivar which is included in this group. And you can see in the red box, which is the Saprothera leaf blotch across the crop management in some of the ranking Shukla stability variance, which is the most important predicting factor in case of in both managements. So its value is higher than 7. So if it's near to the 9, so its mean is more stable. So it's very in both managements Saprothera leaf blotch. This disease is the more predicting important factor, which is tell us is stability. And in the multi-trait stability index, you can see stem rust, which has the greater than and equal to 7.7 value, which is also close to the 9, which is the more stable. And you can see there the stability of the cultivars. and the high input crop management in the Shukla stability variance. In the second line, you can see the fully ripe days, which are lower than 200. So it means if they have lower than 200 fully ripe days, it means this cultivar is more stable. So it's a type of indication which tells us about the stability of by using this disease resistance and these stability analysis. So this table is also showing the ranking of Shukla stability variance. The upper table is moderate input and lower table is a high input crop management. And we sum all of these ranking in sum. You can see the sum column which showing the different values. The lower value means more stability and higher value means lower stability. So these both management levels, you can clearly see the difference of the performance of these cultivars is different. So and their stability is different. So this table is a multi-trait stability index in both crop management, and you can see the ranking value, which is first mean is the more stable, and the higher value of ranking mean is less stable. So in both crop managements, different cultivars told us the different stability. So their performance is different in both kind of crop managements. So finally, we conclude our presentation. So in our work, hybrid cultivars showed higher yields as compared to the population cultivars. And the protein contents of the hybrid cultivars were lower as compared to the population, is about 3 to 6.5% lower in moderate and high input crop management respectively. So we didn't find any significant difference on the influence of yield components on the yield condition between these hybrid and population cultivars. But we found that the number of spikes which have the higher influential factor on the yields. And we also propose a simple sum of the Shukla stability variance ranking simple parameter approach, which told us about the stability across the various traits simultaneously. So it's a very simple approach which we assess in our experiment. So these results indicate the cultivation of the rye can contribute to integrated pest management, and we can reduce our input management by these stability analysis, and also can improve our overall dietary nutrition. So these factors are very essentials to ensure and enhancing the food securities on a broader scales. So I would like to thank you very much for your attention. If you have any questions, please.
What is the practical application for this result?
Yeah, it's very practical. It's very nice question. Thank you. Especially for the farmers and the industries which are interested in different kind of quality parameters. So they can choose which kind of cultivar is can well perform by using this minimum of resources and minimum uses of these nitrogen fertilizers. So they can also use, and also we can recommend to the farmers. So they can increase their profit by minimum expenses on the fertilizers. Thank you.
Okay, thank you.
So I think that's all questions. So thank you for a very interesting presentation. And next will be Mr. Paweł. Jeszcze brawo. Tak, właśnie. So next will be Miss Zofia Utrikodadi, technologia żywienia i żywności. No
Thank you. I will also speak in English so that everyone understands. So I would like to tell you a couple of words about the fish intake among Polish youth, its determinants, and complying with fish consumption recommendation. So this is the agenda for my today's speech. And in the beginning, a couple of words about why it is of importance to assess fish intake and the recommendations. So as many of you might know, but maybe not all of you, fish is a very good source of protein in our diet, but not only protein, it's also vitamin D, iodine, selenium, and omega-3 fatty acids. But the most important ones are vitamin D and omega-3 fatty acids because they are not found in many other food items. That's why eating fish is important concerning those two. Meta-analysis show that an increase in fish intake lower the risk of many diseases such as the metabolic syndrome, dementia, depression, but also heart attack and stroke. That is why international and national organizations and associations recommend to consume at least 2 portions of fish weekly. One of those portions should be fatty fish because fatty fish contain more omega-3 fatty acids and more vitamin D. And this is around 200 to 300 grams of fish weekly, depending on the portion size, which is different in different recommendations. But how much fish do Poles consume? We don't really know. We have some data from the household budget service, but it actually doesn't show individual data. It shows the fish consumption in the whole household, also including animals, but also the food we throw away. We have some data from balance sheet data, but this actually shows more what's on the market than what is actually eaten individually. We do have some studies which try to assess fish intake, but the method used in those studies, such as, for example, the one you can see here, Wabash, they use the 24-hour recall, which means that they ask people, what did you eat yesterday? And since we usually don't eat fish every day, if it was not Friday, let's say, probably many people didn't say that they ate fish. However, if they ask about Friday, which is typically a day we eat fish, it might be more than we actually do eat. That's why we don't really know. We also know from other studies conducted in other countries that usually younger people, have a lower fish intake than people in older age groups. We also know, probably you also have this view that nutrition in childhood and early adolescence has consequences on the proper development of numerous systems. So it's very important. And adolescence is also the time when dietary habits are created, which often persist in adult life. Therefore, it's so important to assess the dietary intake among young people, because this is the time when dietary habits can also be influenced, changed, and this is how people will probably consume food later in life. That is why the aim of the study was to assess fish intake in a nationwide sample of Polish youth, as well to assess its determinants, so what influences it, and to assess complying with fish consumption recommendations. A couple of words about the material and methods so that you understand the study better. The study was conducted among more than 1,300 adolescents. They were aged 14 to 22 years old. And those were people from secondary school students from all regions of Poland. We included 5 different types of secondary schools because we aimed at having a representative sample of young people. However, as you can see, looking at how many female and male we had, we didn't manage to get a representative sample because, of course, taking part in the study was voluntary. So despite choosing a representative in a representative way, the schools, the answers we got were not representative. However, we got quite a lot, as you can see. It was a cross-sectional study, so we asked the students just once at one point of time, and we used the Cowie method. We assessed the fish intake using a food frequency questionnaire, which was based on a validated short food frequency questionnaire. And we assessed the fish consumption of the participants, of the students, but also the fish consumption of their parents or legal guardians. We also used a nutritional knowledge test about fish, which we based on a study by Berger and Gochfeld. Here you can see the statements, and the participants were asked to say whether they think it's true or false. And this is how we assessed what they know about eating fish. And of course, we also asked about some basic data. Let's go to the main results of the study. This is a table showing the fish intake in the studied group and also in different subgroups. I hope you can see that. Yes, I think you can. So the median fish intake in the group was 35 grams. And just to remind you, the recommendations say it should be at least 200 to 300 grams weekly, so it's more than five times less than the recommendations, which is quite alarming. The fish intake did differ among different subgroups, and it was higher among males. It was also different among students going to different schools. Interestingly, it was higher among those who go to comprehensive high schools. In Polish, we call those liceum ogólnokształcące. So it was higher among those students than among those who go to technical or vocational schools. The place of residence, the region where the students live, did not differentiate the fish intake, which was for us surprising, but maybe since the fish intake is quite low among most, well, it didn't differ. The size of the place of residence did also not differ the fish intake. Neither did the body mass index of the participants, nor the age group. Interestingly, and very importantly, fish intake was 2 times higher among those participants who know fish intake recommendations. Those who know that it is recommended in general to consume fish by children and adolescents consumed 40 grams, so a bit more, but those who know the recommendation that fish should be consumed at least or more than once a week, their fish consumption was 58 grams, which is still low, but higher than the other subgroups. We also found a correlation between the students' fish intake and the fish intake of their parent, which you can see here. This table shows the fish intake of the student and the female parent, but we observed the same for the male parents. And this is an important result. Only 6% of the whole studied group met the recommendation to consume at least 300 grams of fish weekly. To sum up and to conclude, the median fish intake among the Polish youth was 35 grams a week, while the recommendation to consume at least 300 grams of fish weekly was followed by only 6%. Males consumed more fish than females, and students from comprehensive schools more than those from vocational schools. And the 2 main determinants of fish consumptions were parents' fish intake and the nutritional knowledge about fish. So this shows that it's very important, first of all, what the parents, what the guardians eat, because often this is what the children eat, and also the knowledge. So knowing that what is inside the fish and how much is recommended is connected to how much fish we eat. And 3 conclusion sentences. From the study, we see that fish consumption among Polish youth is, in most cases, inadequate. And since knowledge about fish is positively associated with fish consumption, nutritional education about the health benefits of consuming fish might be a very good tool to increase fish intake in this group. But the nutritional education should also be addressed to the parents. Here you can see some references. And thank you for your attention.
So thank you for your very interesting presentation. So now we can go to discussion. Somebody have a question? Yes.
Okay, thanks for your presentation. I have a question. Do you maybe have an idea how to change this tendency? You thought about it?
Yes, I think what we should do, but maybe also fish producers should do, is to teach people more about the good sides of eating fish rather than scaring people off eating fish, because this is something we can see is happening. Some documentaries are being shown to people, showing the bad sides of fish or about the bad sides of farming fish, about some pollutants that might be in fish, which is true. But nowadays, many food products do contain some contaminants. You have microplastic in bottled water, you have pesticides in strawberries. It doesn't mean we shouldn't drink water or we shouldn't eat strawberries, nor about the fish. The fact that they might have some antibiotics or that they might have some microplastic, according to science, the health benefits outweigh the potential risks. So teaching people more about the benefits and maybe telling them that, okay, yes, they might have those dangerous or unhealthy elements, doesn't mean that they are not healthy. Yeah.
You have a question, Zosia? Is there any difference between omega-3 content between raw fish or fried fish? Like
fried?
Yeah, fried or oven.
Mm-hmm.
Is there any difference?
Yes, there is. If you bake it in the oven and don't add any oil, you just get rid of some of the water, so actually the percentage increases. If you fry it in oil, some of the oil gets exchanged. So the oil you're frying in goes into the fish. So in this case, it's also important which oil you use for frying.
Could you tell us something about the price of fish? Is it a barrier in Poland or not?
It definitely is, or is often said as a barrier. However, we do have different fish species, and a fish species I often recommend is herring, which has a lot of vitamin D, which has a lot of omega-3, and in Poland it's much cheaper than salmon, for example, or a different example would be rainbow trout. This is the fish which looks like salmon, but is not salmon, and this is often quite cheaper. So yes, it is a barrier. However, looking at what different things people consume, such as snacks, chocolate bars, chips, I think it's an excuse that strong tęczowy. So looking at how much money we spend on food we don't really need, so the food we just eat for pleasure, I believe that it's still possible to include fish in the diet. And even without having a very big budget for it. But yes, people do say that I don't eat fish because it's expensive.
Do you have your own thoughts about why the consumption is higher in males than the females?
Yes. Actually, my hypothesis was that female consume more because the way we see females stereotypically is that females look more at healthy food and like men prefer meat and fish. But actually, it's probably just because of the portion size. If a man eats fish, it's usually a bigger portion than a woman. So the median then at the end, like the average is then more in a week, probably because of the portion size.
Thank you.
Thank you very much. Wracając do dyscypliny nauk biologicznych, mam przyjemność przedstawić Julię Nowakowską, która zaprosi nas do świata zawiłych mechanizmów układu odpornościowego i zagłębi się w temat innowacyjnej terapii glejaka wielopostaciowego. Zapraszam.
Good morning, everyone. My name is Julia Nowakowska, and I'm a PhD student at Center of Cellular Immunotherapies. My supervisor is Professor Magdalena Król, and today I would like to present you how we can unlock glioblastoma defense using macrophage drug conjugated therapy invented by our laboratory. Nowadays, we know so many different types of tumor, of cancer. However, why do we focus on glioblastoma? First of all, that's because every year, nearly 300,000 of patients are diagnosed, and from this group, over 240,000 die because of this disease. And even though nowadays we have treatment methods like tumor resection, chemotherapy, and radiotherapy, the average length of survival of these patients are only 8 months. But when we think about cancer, we mostly have in mind only cancer cells. But it's crucial to know that cancer is not alone in our body. It's surrounded by our own immune cells, which comes to the tumor area, to tumor microenvironment, to kill the cancer. However, sometimes these tumor cells could be stopped by regulatory T cells around the cancer area, and they cannot kill cancer cells so effectively. These tumors are really hard to cure. On the other hand, it could be the situation when, for example, macrophages, dendritic cells, and lymphocytes can enter tumor area and kill cancer cells effectively. So, having in mind that macrophages can enter tumor area and cure the cancer, our laboratory invented therapy based on macrophages. So these cells are conjugated with a drug, and this is how this shortcut comes from. This is MDC, macrophage drug conjugates. So macrophages naturally migrate to the tumor area, and in a process called phagocytosis, they start to kill cancer cells. At the same time, they deliver anti-cancer drug, which makes them killing cancer cells even more effectively. We tested this mechanism in vitro, and it gives us the permission to test it in vivo as well. So mice having brain cancer, we treated by our macrophages conjugated with a drug, and they survived much longer than other controls treated without macrophage drug conjugates. This is why we decided to take the next step and re-challenge the tumor in the mice who survived. It ended up that after treatment and after again tumor implementation, the tumor did not grow. So it gives us the clue that somehow these results shows that MDC, macrophage drug conjugates, may provide an excellent therapeutic option for patients, for humans. So we decided to check this mechanism more in detail and check how this therapy influence our immune system. So these cells, which I previously shown you in the tumor microenvironment, how they are influenced. It ended up that B cells and T cells there are more B cells and T cells in the tumor microenvironment after treatment with macrophage drug conjugates, which shows that somehow our immune system is stimulated to cure cancer on their own. Moreover, further studies shown that these T cells, T lymphocytes, are also activated so they can kill cancer cells more effectively. That is why during my PhD studies, my job is to check how these macrophage drug conjugates can interact with dendritic cells and how can it influence cancer treatment. That is why during my research, I did co-cultures with cancer cells and with our macrophages loaded with a drug. At the same time, I differentiated dendritic cells, and then after taking conditioned media from co-cultures, I checked how they influenced dendritic cells. But firstly, I had to get the research material, and it is really convenient that I can isolate it from patients' blood. Starting with Buffy coats derived by humans, this is the human blood. After a series of centrifugation, we can isolate the mix of peripheral blood mononuclear cells, where inside are macrophages. But after separation on magnetic columns, we can isolate macrophages. And then after adding specific interleukins and growth factors, we can differentiate them into dendritic cells or into macrophages. So as a result, I had the material to provide my research, my studies. However, differentiating dendritic cells ended up being quite challenging. So I had to optimize the culture media I cultivated the cells in. So it ended up that addition of 10% of fetal bovine serum gives us the best results of having the most live and cells in a good condition, so they could be used for further studies. Here on this left picture, we can see a lot of tiny elements. So these are the parts of dead cells. However, here, after addition of fetal bovine serum, all the cells are in a good condition because they, for example, try to attach one to another to communicate together. This slide shows it more in numbers. So after when I cultivated cells with no FBS, the recovery was really, really low. So I did not have enough cells for further experiments. But after addition of FBS, this recovery was really, really satisfying. It always happens that some of the cells will die. However, here I had enough research material. And this last graph summarized that. As we can see, after addition of fetal bovine serum, this cell's recovery was significantly higher, and the cells were in a good condition to perform further studies. This further study is the experiment summarized here. I decided to show you pictures how the cells look like when we add conditioned media to dendritic cells. And this is the control where there was no conditioned media. So we can see in every case, cells changed somehow. And this is the point of my research, to see how these cells are changing. However, now I can show you only this on pictures because I started my PhD this year, but in the next months, I plan to investigate this phenomenon further, and I hope in next year present you more detailed research. So thank you very much for your attention. And if you have any questions, I'll be happy to answer.
Okay, thank you for your great presentation. I don't know if I've understood everything correctly, but you've said that like this technology is in vitro in the cell cultures, but also you've tested it in animals.
Yes.
So do you have any plans, or it is possible already, or maybe not, to go with it further to the like clinical trials, or not yet?
Yes, and actually, that's the plan for to go with this results to clinical trials. Only I can say it would be really soon. So
That's great. Thank you.
thank you.
It's a great presentation, but I have one question. You mentioned phagocytosis in mice increase the lifespan of the mice. But do you think it can completely stop the cancer for further development, or it can just can stop for a few don't to flourish further, or we can say it's a completely cancer free animal, or we can just say it cannot develop further. Yeah.
So in case of glioblastoma, the metastasis, the risk of metastasis is quite high, but for now, I think we can say that if the tumor does not reoccur again, because the immune system reacts for that, possibly also it will react if the cancer would occur again. Did I answer your question? Because the cars were noise around here, and
I can repeat my question again if you didn't. Can we say the mice which you treated with the phagocytosis phenomena with your drug, they increase the lifespan of the mice. So can we say it's a completely cancer-free animal or not, or it can just stop the further development of the cancer cells?
So I think saying it's totally like cancer free would be too much, because even if we consider a patient cured, we never know. There is always a risk if the tumor will reoccur, if you have the malignant tumor once. However, this therapy shows the results that suggest that our immune system can defeat the tumor. So, yeah.
Thank you.
Thank you.
Thank you very much. Przejdźmy do następnej prezentacji. Problem marnowania żywności na pewno wielu z nas wzbudza ogromny niepokój. I sprawdźmy zatem, jak wygląda ten temat z punktu widzenia ekonomii i finansów. I zapraszam do zaprezentowania się Sylwia Devi i Sagita Andik. Może brawa na zachętę.
Let's pointer.
Okay. Good morning, ladies and gentlemen. My name is Sylvia Dewi Sagita Andi. I am PhD student from the Institute of Economics and Finance. And today I would like to present my working research paper and entitled Tackling Food Waste in Urban Landscape, a systematic review of household food waste in ASEAN countries. So this is the authors are Sylvia is me and Joanna Rakowska is my supervisor. So what's the background for this research paper? So as we know that over 10 billion people by 2015 is expected to intensive demand for food. And now we already have limited resources. So it is the main problem today. And then concurrently, global food system also face some challenges. One of them, as we know, that the climate change. And from the Tomlinson and Clark et al. Revealed that one of the big contributors to climate change is the escalating issues of food waste. In addition, urbanization also has impact to the increasing of the food waste. It's also revealed by some researchers. And as we know, maybe in the urban areas or in the Warsaw or metropolitan areas, we have different lifestyle. For example, the worker people or the busy people tend to order food through delivery order service. So they buy the food outside and then bring to the home, and maybe the portion of the food is not appropriate with their needs. So it tends to generate the food waste at household level. So I highlight household level, which is the food that didn't consume in the outside but at home. Then, in fact, the dominant food waste production is produced at the household level. It's not only in the developed countries, but also in the developing countries, most often in ASEAN countries. So I will to give you what's the countries in ASEAN that produce the highest or huge number of household food waste. That's why this research will be fill the gap because only a few studies have been conducted to investigate the factors or antecedents that contributing the food waste at household level in urban areas. So specific in urban areas, especially in ASEAN countries. Yeah, this is the objective. This is the qualitative approach. So the first, the systematic review attempts to bridge the current knowledge gap by the conducting comprehensive analysis household food waste in urban areas. And the second, to examination of antecedents factor associated with the food waste in urban household. And then the last one, to identify successful strategy, intervention, and policy implement in ASEAN urbans to tackle the food waste. Yeah, I use qualitative approach. So there are some criterions. The first one, information source and search strategy. So this study retrieved database from the two database, Scopus and Web of Science database from January 2018 until June 2023. So the last 5 years. Why those of database? Because we think that it's more reliable and accurate for an update also. And for eligible criteria, we have 4. The first, it should be journal or article, not proceeding or not the book. The second, the article is written in English. The third, the research article is social science. So in the Web of Science and Scopus, we only tick the social science. And then the last one, the article related with the food waste at household level and urbanization or urban areas. And how to extract the data and synthesize. So we are referred to the PRISMA analysis. It's common use for the most researcher in the last year or the last year. It's for to make a systematic review analysis. So we call it PRISMA. In the PRISMA, prefer reporting item for systematic review and meta-analysis. There are 27 item checklists that we should to fulfill the criteria. This one, the result, the first result. This is the picture in the right side. This is the flow of the process to retrieve the data or article review and study selection. So, as I mentioned before, from Scopus and Web of Science database, there are 3 stages: identification. In the identification, we use these keywords for the Scopus and Web of Science, and then we retrieve around 7,000 articles, and then we exclude the duplicate article. So, and then we exclude the non-text English article, and then in the screening, we get the around 5,000 articles, and then we exclude the irrelevant titles and abstract, and then non-review article, non-ASEAN countries, and then article did not investigate between household food waste and behavior urbanization. And then we get 41. In addition, we also add some literature review article from the citation and searching. So the total review 41, but we only review 31. Why? Because some of the paper didn't provide full information. For example, maybe it's ASEAN countries, but didn't mention what's the name in Asia. So we didn't include in the review. So because we don't have enough information for this. And this is the picture that I processed to explain what's the country the most that conducted household food waste in urban areas. So the first rank in ASEAN countries is China. During the 5 last years, China is the first rank. There are 10 times research that conducted about the household food waste in the urban areas. And then 4 times, Vietnam, Malaysia. Yes, Vietnam, Malaysia. And then 3 times in Indonesia, Thailand, and Philippines, and then Iran. And then 2 times Japan, India, Bangladesh, and then 1 time Israel, Saudi Arabia, and Myanmar. Why China? Our assumption is because China has a huge number of population, so it's tend to generate food waste, and also in Indonesia. Okay, but we conclude that based on the Scopus and Web of Science database that there are some countries did not appear in this research that didn't investigate about the household food waste. So also it is important for our research. Then I classify what's the research design approach in the review article that use. Most of them quantitative approach, and then using questionnaire and survey. Yeah, this is the result of synthesis for the 31 review article. So we are classified, there are 5 domains antecedents that affecting the household food waste in urban areas. For the first, household attribute include education, age, income, household size, gender, space of land to compose, and then food expenditure as well. And then the second, psychological factors like knowledge about the food waste, attitude, perceived norm, environmental awareness, subjective norm, safety awareness. And then the third, behavioral factors. For example, if you checking the level of food or not, because the food expire date is also impacts to the to reduce the food waste. And situational factor, food availability, accessibility, COVID-19 pandemic. Why? Because in the research paper also conducted during the COVID-19 pandemic, so it's impact. And then the food attribute, food price, sustainable product, and quality. So this is the conclusion: climate impact of urban food waste. And then the majority of recent research on household food waste has been concentrated in ASEAN countries. And then there are 5 domains that influencing the food waste, and then need for sustainable solutions. So the study underscore important addressing consumer behavior, family dynamic awareness of environmental impact to minimize the food waste. This is the reference. I didn't include all the reference because it's not enough. Thank you for your attention.
So now we can go to discussion and we have first question.
Great presentation. I'm interested in why you chose only social sciences journals. What was the reason for that?
Okay, thank you so much. Yeah, why? Because if I choose some subjects, for example, environmental engineering, it's there are some terminology that's included in the social science. For example, maybe in environmental technology, like gas emission. So it's not our expected gas emission and the impact for the climate change. It's too broad for us. So we just make this restriction only the social science that related with the economic and social. Focus on that, like behavioral.
What can do Asian countries to reduce food waste in the urban area? Could you tell us about only two main ways? What do you think?
Okay, in urban areas, the first thing when I read when I summarize this research is the intervention from the government regulation, because we have to force the people to reduce the food waste. If we only talk, please don't do food waste, blah blah blah. It's just yes. They didn't, yes. It's just a reminder. But the regulation, for example, in Indonesia, Bogor city, we have regulation. If you it's like it's like about the money incentive at the first. Give incentive if you can reduce the food waste or make compost. There is incentive, but not for the all of the areas. But I think it's not continue because it's very expensive.
I have one more question.
Okay.
Did you encounter any differences among those Asian countries that you had data for about the food waste, in which way they?
No, because it's qualitative.
So you didn't look at?
Yes, because I just review all of the article from the so it's qualitative. I didn't do meta-analysis like yes, quantitative. Yes, yeah.
But looking at the qualitative, were there?
But I have the data about it. Some of the review data about the how many weights of the food waste in the household, but not of the country have this data.
Okay, so thank you very much for your great presentation. Thank you. Ten wtyk, czyli ten granatowy, czy ten czarny? Okej. Zatem myślę, że teraz możemy już zaprosić kolejnego prelegenta, reprezentanta inżynierii środowiska, górnictwa i energetyki. Zapraszamy pana Pawła Łochowskiego.
Drobna poprawka, jestem inżynierem lądowa, transport i geodezja. So hello everyone, my name is Paweł Łochowski, and today in the short presentation I have allocated today, I want to introduce you to a topic I'm very passionate about, which are drones and geospatial technologies, particularly the use of unmanned area systems and remote sensing methods in the analysis of urban greenery on the example of Warsaw tramways. The computer has stopped working.
It's coming. A nie przeczytaj, że przełączyłeś tamten?
Od tego slajdu.
So going onward with the presentation, I would like to introduce a research problem. So actually green tracks, why are they important? So they introduce the proportion of permeable and biologically active areas. They increase water retention. They reduce the effect of urban heat island. They improve air quality, reduce noise thanks to the lower vibrations, and have great aesthetic qualities. So I think after this slide, it's quite obvious why green tramways are quite important, but they also have quite a few challenges. So their maintenance is quite challenging. They are subject to many stressors, including high temperatures, low water, and salt. The maintenance operations are very expensive, and their surveillance is very complicated. So as a fun fact, Warsaw Tramways asked MPWiK, which is responsible for water delivery in Warsaw, to deliver water to the 80 kilometers of green tramway that Warsaw Tramways possesses. And MPWiK determined that Warsaw does not have that much water in it to actually give it to Warsaw Tramways. So the research problem is actually quite extensive. So the scope of current and planned studies is, to date, I've conducted studies with the Phantom 4 multispectral with a 2 megapixel spectral resolution camera. But planned studies include NDVI maps from a Mavic 3 multispectral, which is higher spectral resolution of 5 megapixels. Also, thermal resolution is added with the Mavic 3 thermal, and fieldwork has been conducted. The areas of current research are Prosta Street, Aleja Jana Pawła II, ulica Kasprzaka, and Puławska. So these are covered in grass and sedum, but the planned areas include 20 sections in Bemowo, Białęka, and it's pretty much in all Warsaw, from Wionów to Białęka. So as you can see on the map, it's pretty much all over Warsaw. And I've actually completed 16 of the 20 planned sections, so that was me. And if you imagine how measuring chlorophyll content and temperature looks like on Warsaw Tramways, if you ever played Frogger, I did that, but just with trams. So they were quite excited to see me on the tracks and were honking at me all the time. So the input data consists of spectral resolution layers, which I processed into NDVI. NDVI is a well-documented index used in geospatial systems, and it's calculated from near-infrared and red layers. So let's unpack what NDVI means. So it's normalized differential vegetation index. So the normalized part means that it's always from -1 to 1. Differential meaning that we use two layers and they are always the same. Vegetation index, of course, meaning that we are measuring vegetation. So as you can see in the top right, this is the raw value, where 1 is pure white and -1 is black. So what we do as a next step, we classify the data with a discrete classification. So that's this map, where we overlay the -1 to 1 data in a more straightforward way, which is also based on literature. So that's why it's well documented. NDVI is the most popular index in this category. So understanding the raw data is quite challenging. So as you can see in the top right, maybe you might be able to see some tracks, maybe some vegetation, but it's quite challenging to unpack. So we classify it in this way, where the red parts are road, concrete, tracks, sand, then dry grass, bare earth, and then grass and sedum of low and high condition. So that's the green areas in which we are the most interested. And I think that looking at this map as a client, you're quite well versed in what that means. So going into the detail of the data, we can see areas of high and low vegetation health. So for example, here you can see where the tram is going over the green tramway and kind of hitting the grass. So you can see in the map that it's also a darker color, meaning that the health is lower. We can also see patches of grass that are higher health, which is also reflected in this map. The data is so high resolution that we can even see small patches of grass in the pavement, which is of course not intentional, but we can also see them. So the resolution of the data is 2 to 3 centimeters per pixel. So the smallest detail we can resolve is about 2 centimeters. This is the initial result of the survey. So the first table is all of the data. So it includes all the classes. And the most important value are the percentages. But to be honest, looking at this once again as a client, it's quite challenging to figure out what this means. So what we did is we removed the road, concrete, rail, sand class, which gave us just the bare earth, poor and good condition sedum. So that is the result here. And what we can confirm initially with the data is that, as Warsaw Tramways expected, sedum has higher health compared to grass. So that is the initial result. So we can see that it's almost a 10% difference. So the advantage of using sedum instead of grass is obvious in this slide. As a result format for the client, in this case, Warsaw Tramways, we use RGB orthomosaics. So they are an overview, just a standard map of the surface. We have spectral ultramosaics as an intermediate step before NDVI. Then we go forward to NDVI, which is the processed colorful map you've seen on the previous slides. Then classify it, which applies the different classes to the ultramosaic. And then we can pinpoint the locations of low and high health areas, which gives Warsaw Tramways go here to fix the green tramway in this place exactly. As for the results, from the pilot project we have conducted, we can see that we need more sophisticated categorization and classification technology, which we're implementing at this time, which should better separate bare soil and other classes between each other. And further field work will actually confirm what's the correlation between, for example, high temperature and low chlorophyll index of the sedum or soil to, for example, a low NDVI. So then we can actually say what the results are exactly. And with this, I would like to conclude my presentation. Should you have any questions, I'm more than happy to answer them. Thank you.
Some question?
Yeah. I have a question about sedum. I'm not from this area, so I don't really know what's the difference between sedum and grass. So that's my first question.
Sure. Okay, so let's unpack it. So why it's important, it's actually a good question, so I can introduce what actually covers green tramways in the time I did not have, so thank you. So green tramways are covered mostly in grass, but the newer technologies actually include sedum. Sedum is more, how to say it? It's a newer technology which has plants that are more resilient. Mm-hmm. So it's rozchodnik. And it's just more resilient to high temperature and low water contents in the earth. On the contrary, it's more susceptible to salt. So it's kind of a trade-off, and Warsaw Tramways are introducing that to see what is better. Mm-hmm. The newest cover of green tramways is actually ruderal vegetation, including oregano. So it's just a mix of different plants that are very resilient, but also, once again, trade-offs. So next time when you're going on a tram and you're quite hungry, you can think of a pizza and oregano, and then, you know, that's a trade-off.
Great, thanks. And I have one more question about the water. That you said that is actually we don't have enough water to water all the grass or sedum in Warsaw. Is the rainwater not enough, or
are there no plants that we could plant so that the rainwater would be enough?
So the answer is no, we don't have enough water. And actually the initial point of Warsaw tramways, because the issue is that water, when it rains, it's not collected, it just goes down into the soil. So it's actually not collected and used by the grass. It's, there for a few hours and then it goes into the deeper parts of earth, because green tramways are not a natural part of soil. They're not having the same processes that you would have in nature. So initially they wanted to build kind of reservoirs for water to store it after rainfall and then put it on the tramways. But it was so expensive. I don't remember the scale, but it was just such an expensive project. It was so infeasible that it's like kind of out of proportion. So the next idea was, okay, let's ask MPWiK to deliver us, this many liters of water between 1 a.m. and 4 a.m., which is like the optimal time to water plants. And they're like, yeah, well, we don't actually have that much water in Warsaw. So yeah, that was the challenge.
And based on your research, what is exactly your recommendation for Warsaw tramways? How to increase the vegetation, or what is your recommendation?
So we have a strategic part to this presentation. So we are preparing a framework. We're working very close with Warsaw Tramways, and they are quite excited to work with us because we can point them to some results, and they are very eager to use the results of this POC to kind of introduce them into the real world. So that's very nice for me because I actually feel that my work is kind of being introduced. But what we see in the results is that we can detect areas of high and low plant index, which we can then tell Warsaw Tramways, okay, for example, this area of Warsaw has high cover with high buildings, like you can see in this slide. This is Rondo NZ. So here's like Złote Tarasy, here are really tall buildings. So for example, this area would be perfect for grass because you have cover, you have quite a lot of buildings, so there is shade, there is lower temperature. Okay, this is a good place to use grass. But then we have other places where instead we can tell them, okay, the temperature in this part is very high, so let's use sedum here. But for example, this part is very close to the road, so maybe ruderal vegetation would be nice here because we have road salt. So we want to propose them, okay, for each area use a different plant, and also they spend a lot of money to keep up the vegetation. So we can actually check before and after. If they tell a company, okay, perform some kind of maintenance on the part, then we can check, okay, actually it improved the Warsaw Tramway or there was no change. So what they did is actually meaningless. So that's the answer. Thank you.
Thank you.
Dziękujemy pięknie. Teraz czas na małą ciekawostkę, bo dowiedziałam się ostatnio, że Polska jest 3. światowym producentem jabłek. No i nie bez powodu są to też jedne z ulubionych owoców SGGW. No i dlaczego o tym mówię? Bo z jabłkami wiąże się też temat następnej prezentacji. Więc wróćmy zatem do dyscypliny nauk biologicznych i serdecznie zaprośmy do prezentacji Marcina Tymińskiego.
Okay, thank you very much for having me and thank you for this nice introduction. Today's my presentation will be about the profile of carbonylated proteins during actually dormancy alleviation in apple seeds. So first, I want to catch up with you about some basic seed biology terminology. So what is the seed dormancy? It's actually a state of temporal block of germination. So the seed is vital, but even under a favorable condition, it won't germinate. Then the germination is the process of activation of of embryonical metabolism that leads to seedling formation and seedling developing. And actually, this is quite interesting that germination ends in the very moment when embryonic groove cuts through the seed coat. So after that, we are talking only about the plant growth. So during my presentation, I will be focused only on this dormancy and germination here. So there are some procedures to remove dormancy from the seeds. One of them is the seed stratification that relies on mixing seeds with sand and storing that in various temperature for optimal time. In case of apple seeds, actually we called about cold certification because the temperature that is required is 4 degrees Celsius, and we store that seeds up to 40 days in the fridge in this temperature. So actually during dormancy removal, there are many metabolical changes that are taking place. One of them is the formation of reactive oxygen species. So those are the chemicals that are highly reactive due to their chemical nature. And in optimal concentration, they play a role of cellular signalers. But when their concentration is too high, they actually can alter the proteins, lipids, or nucleic acids, changing them and leading to loss of their biological function. On the other hand, the optimal concentration of reactive oxygen species is incredibly important for seed biology because only this optimal concentration leads to dormancy removal and germination. Too high concentration of reactive oxygen species leads to cellular damage, as I've already told you, but not enough does not have any effect. So, as I've told you that reactive oxygen species might alter, for example, the proteins, the actual modification is called the protein carbonylation, and it relies on adding the carbonyl group into the proteins. And actually, such proteins might be later degraded via ubiquitin proteasome pathway, creating the peptides. The peptides later might be also degraded into amino acids that might be used for the embryo to grow. Because you should know that at the very first beginning and the very first stages of germination, embryo does not produce its own amino acids. They are not synthesized yet. So the embryo relies only on those materials stored, for example, in proteins, in case of proteins. Therefore, the aim of my work was to identify the proteins that were carbonylated during dormancy alleviation in apple seeds. So I've conducted the stratification procedure for 7, 14, 21, and 40 days in the conditions I've told you before. Sorry. After that, the embryos were isolated from the seeds, and from them, the embryonic axes were isolated. And I've conducted my experiments solely on those embryonic axes. So what I've done, I've isolated the proteins and then separated them via two-dimensional electrophoresis. So I separated them through their mass and through their isoelectric point. Then I've used the Western blot technique to visualize the carbonylated proteins. I've selected those proteins and cut them from the gel, and then digested it with trypsin. And such digested proteins, so I've got peptides, I've analyzed them using the mass spectrometry with the Malditov technique. So now I want to briefly tell you a few words about this technique because I think it's quite interesting and maybe some of you might want to use it someday. So as I've told you, the sample that you acquire is mixed with matrix, placed on the steel target, and then inserted into the mass spectrometer. Then the laser fires up, actually, and the ions are generated. Those ions move through the electric field up to the detector that detects their mass and their charge. In the same time, their time of flight is also detected, and as a result, we've got the mass spectrum. And it's quite important for you to know that each protein and each peptide may give different and very unique peaks pattern in this mass spectrum. So then the software analyzes this mass spectrum and compares the results with that database. And then we got the list of identified proteins in that sample. So moving to the results, I want to briefly show you the proteins profile in embryonic axis during the dormancy removal. And as you can see, as stratification procedure progresses, there are much more less weighted proteins in the gels comparing to the very first stages of dormancy removal. Then I've done the image comparison and I've selected those carbonylated proteins that I've later cut from the gel. And I've got a very long list of identified proteins. So I'm only showing you the selected part of it. The list is very, very long, but I was able to find some groups that occur more often during this in my samples. So one of them is the late embryogenesis abundant proteins. Others are the seed storage proteins, heat shock class proteins, seed biotin containing proteins, or some stress proteins, low temperature induced stress proteins. So let's briefly talk about these groups, these families that I've identified. So the sole purpose of seed storage proteins is to being degraded because they are the storage for the carbon and nitrogen in the seed. And as I've told you, in the very first stages of germination, embryo does not synthesize its own amino acids. Then we have the seed biotin containing proteins that actually store biotin, that is a very important cofactor for some enzymes. And those families are also part of the late embryogenesis abundant group that helps seeds, helps cell go through the desiccation, because you must know that during seed development, there is a very big water loss in the seed. The seed is dehydrated. So those proteins help the cell protect against the water loss. Then we've identified the heat shot proteins that are also the proteins responsible for the response in various stresses, not only heat shot but also water loss, drought, and other temperature shock. There are also some kind of protein chaperones, so they are helping other proteins to keep their third and fourth structure in touch, to being folded. And last but not least, I've identified some low temperature induced proteins that also have some impact in stress response in seeds. So to quickly sum up my presentation, the dormancy alleviation results in alteration in proteome composition, and the protein families that are actually carbonylated are the storage families and the stress response proteins. And it's very important that we've come up with this identification, because this may help us fill the gap and understand the process of seed dormancy alleviation better. Thank you very much for your attention.
Bardzo przepraszam, zasłuchałam się do tej wybłuszka. Czy są jakieś pytania? A może jakie są pytania?
Can apple producers use your results for better storage of apple seeds or not really?
I mean, those are quite basic research. And actually, apple trees do not reproduce. I mean, in the orchard, we are not using seeds to reproduce trees, actually. We are using different techniques. So those, we are talking about apple seeds because they're a great model organism for the research for dormancy alleviation and germination, because they are, I haven't told you, but they are kind of the orthodox type seeds. So they are actually very, very dehydrated. And most of the seeds around the world are also at this type. So that's why we are using this model.
Thank you.
I may have maybe a silly one. If I would like to grow a plant from avocado seed, can your research tell me a bit how should I reproduce them so I would get the best avocado tree?
I mean, this is, as I've said, some kind of basic research, so not exactly. But in case of avocado seeds, I mean, the seed coat is also kind of a problem because it's very, very thick, and you have to like somehow the water have to go inside the seed, as you know. So, for example, this is the same case in apple seeds. They also have the seed coat, so we have to somehow overcome that to affect the embryo to germinate. So in case of, for example, avocado, I would say that just placing it in like in the gas or in the water is the good solution.
Okay, thank you.
That's the second question.
Thank you for your great presentation. My question is about your methodology. Is it possible to apply that methodology to detect the CO2 emission from the soil or some kind of things?
Sorry, what seed emission?
Yes, that methodology you used.
The Smalditov technique.
Yes, yes. Can we detect the CO2 emitted from the?
No, actually, it's not this this kind of technique. This technique might be used to identification of the proteins, or for example, the nucleic acids or lipids only. So we are not detecting the basic compound like CO2, but we are looking at more like biological structures, biological chemicals.
Thank you very much.
Okay, thank you very much.
So now we have the last speaker, and this is Mr. Manoy Kumar Yena from Hort Agriculture Institute.
Welcome. Good afternoon to everyone. Myself, Manoj Kumar Jena, PhD student in the Department of Plant Protection. Myself, Manoj Kumar Jena, PhD student at the Department of Plant Protection in the Institute of Horticulture Sciences. Now I am going to present about my current research entitled "The Fast Record of the Effect of Humidity on the Functional Response of a Predatory Soil Mite." According to Solomon, functional response is the number of prey successfully attacked per predator as a function of prey density. Holling classified functional response into three types, such as type 1, type 2, and type 3. In type 1 responses, the number of prey eaten increased with increase in prey density up to a certain density and then remains constant. Whereas the proportion of prey eaten initially remained constant up to a certain prey density and then decreases. In type 2 responses, the number of prey eaten increases with increase in prey density hyperbolically, whereas the proportion of prey eaten decreases with increase in prey density. In type 3 responses, the number of prey eaten increases following a sigmoid pattern, whereas the proportion of prey eaten initially increases and then decreases. Functional response is an important aspect of predator-prey interaction and is crucial for dynamics of populations and communities of animals. Functional response can be affected by temperature, humidity, age, sex, age and stage of the predator, hunger level of the predator, pesticide, size of the predator and prey, stage of prey, and the host plant. Blattiosus malli is a potential biological control agent of some acarid mites such as Tyrophagus putrescentiae and Tetranicus urticae, and the insects such as eggs of potato tuber moth, thrips and nematodes, and thrips and also nematodes such as Rhabditis scandica. The global atmospheric temperature is increasing that leads to alterations in precipitation. Global warming and alterations in precipitation can affect soil temperature, soil moisture and soil humidity. Fluctuations in humidity can impact biochemical, physiological and behavioral changes in arthropods, including their functional responses to prey. However, there is no report on the effect of humidity on the functional response of the predatory mite. Therefore, I investigated the functional response of Blattiosus malli foraging on its prey, the acarid mite Tyrophagus putrescentiae, at different levels of humidity ranging between 33% and 92%. In this experiment, the functional response of Blattiosus malli was investigated using the eggs of Tyrophagus putrescentiae of different densities ranging between 10 and 160 eggs at various humidities ranging between 92% and 33% at a constant temperature of 25 degrees centigrade and photo period of 16 hours of light and darkness over a period of 24 hours. After 24 hours, the predator was removed and the number of prey eaten was counted by excluding the remained eggs. If in any case the predator was died or escaped, the observation was excluded from the experiment. In this, the functional response data were analyzed in 2 steps. Initially, I determined the type of functional response. Then I determined the parameters of functional response. I determined the type of functional response using 2 methods. One is logistic regression method and another one is using the general version of the Holling disk equation. In logistic regression, according to logistic regression equation proposed by Giuliano, if the linear coefficient is not significantly different from 0, then it will be type 1 functional response. If the linear coefficient is significant and negative, then it will be type 2 response. If the linear coefficient is significant and positive and the quadratic coefficient is negative, then it will be type 3 response. According to Holling disk equation of real, in case of type 1 functional response, the scaling component and handling time are equal to 0, and in case of type 2 responses, the scaling component is equal to 0, but the handling time is greater than 0. In type 3 responses, both the scaling component as well as handling time greater than 0. Then I determined after determining the type of functional response, I determined the parameters of functional response using 2 models, Hassel's model and another one is Cabello and his colleagues model. According to Parvez and Omkar, a predator having a high attack rate, low handling time and high potential of mortality is more efficient. logistic regression results revealed that the effects of humidity, density, as well as their interactions are significant, indicating that the functional response of the predator Blattisucius malli not only affected by humidity but also by the density of prey eaten. The figure showed that the number of prey eaten was increased with increase in humidities. Within each humidity, the number of prey eaten was increased with increase in prey densities. According to logistic regression, the linear coefficient was positive and the quadratic coefficient was negative for 52, 72, 82 and 92% relative humidity, indicating type 3 functional response at these humidity levels. But at 33% relative humidity, both the linear and quadratic coefficients are positive, making it difficult to determine the type of functional response at this humidity. Therefore, I used another method to determine the type of functional response at 33% relative humidity. According to real model, the scaling component is equal to 0, but the handling time was greater than 0 at 33% humidity, indicating type 2 functional response at this humidity. However, at higher humidity levels, both the handling time and scaling component are greater than 0, indicating type 3 functional response, which is similar to the findings determined by using logistic regression. The functional response curves indicate a hyperbolic pattern at 33% relative humidity, indicating type 2 functional response at this humidity, whereas at higher humidity levels the functional response curves displayed a sigmoid pattern indicating the type 3 response. Based on Hassel model, the number of prey eaten was increased with prey density following the hyperbolic pattern, indicating also the type 2 functional response at this humidity. Whereas at other humidities, the number of prey eaten was increased with increase in prey density following a sigmoid pattern, indicating type 3 functional response at all these humidities. Based on Hassel model, the handling time was the lowest at 92% relative humidity, indicating the highest efficiency of predator at this humidity. Based on Cabello model, the potential for prey mortality was the highest, whereas the handling time was the lowest at 92% humidity, indicating the highest efficiency at this humidity. From this study, I concluded that humidity could affect both functional response and the predation rate in a soil mite, and also Blattisucius malli was the most efficient in reducing prey density at higher humidities. Humidity might play an important role in stabilizing the predator-prey system by shifting the type of functional response. Further studies are required to study the effect of other abiotic factors on the functional response. Then these are the references. Thank you.
Okay, so now we can go to discussion. Maybe somebody have some question. No? Okay, Zofia.
I'm interested in how counting of the praise looks like. Like, how do you do it?
I observed the number of praise under the microscope.
And you just count
As
them one
praise
by
are
one.
very small, so that is very difficult to count, but that is possible for
Okay, so you just count one by one.
Yes, one by one, very difficult.
Wow, okay.
Any question more? That's all? Okay, so thank you for your great presentation and bravo.
Thank you so much.
Yes. So the second it will be dinner, but the winner we know at Sunday. Yeah, it's official gal and the best presentation get a prize. Yeah.
To może jeszcze raz gromkie brawa dla wszystkich prezentujących. I teraz chciałabym poprosić o zabranie głosu panią dyrektor Szkoły Doktorskiej, Justynę Franc-Dąbrowską.
Szanowni Państwo, pięknie dziękuję za wszystkie prezentacje, za Państwa zaangażowanie. Tak jak powiedziałam dzisiaj rano, poza oficjalną częścią, wszyscy Państwo jesteście zwycięzcami, pięknie się zaprezentowaliście i jestem z Was wszystkich bardzo dumna. I'm very proud of you. I może dokończę w języku polskim, jest mi łatwiej. Chciałabym również bardzo gorąco podziękować członkom komisji, którzy zgodzili się być arbitrami i ocenić tak gromkie brawa. Ja za chwilkę wręczę certyfikaty. Tak jak zostało wspomniane, wyniki będą ogłoszone w niedzielę w trakcie gali Tygodnia Szkoły Doktorskiej. Bardzo gorąco proszę Państwa o uczestnictwo. Dzisiaj po przerwie, zaraz jeszcze tutaj koleżanki tak powiedzą, będziemy się ponownie widzieli. Ja chciałabym zaprosić tutaj po odbiór certyfikatu członków komisji pani profesor Małgorzatę Kosicką-Gębską. Panią doktor Kingę Noras, pana doktora Tomasza Żelazińskiego, panią magister jeszcze Aleksandrę Jakimiuk, niedługo zapewne pani doktor, i panią doktor Agnieszkę Otłowską. Bardzo dziękuję Państwu za tę aktywność i uprzejmie proszę o ocenę naszych doktorantów. To nie będzie proste. Może pozwolę sobie tu z Państwem. Jeszcze może pamiątkowe zdjęcie. Chodźmy może tak, może chodźmy pod słusznie. Ja już odłożę doktorat, zrobimy mikrofon. Odłożę mikrofon, uwiecznimy tutaj zdjęcie i może za chwilę zaprosimy też wszystkich Państwa. Będzie niezmiernie miło nam tutaj wspólnie tę fotografię przygotować i idziemy na zasłużony obiad.
Jutro ten rogów miała Pani przypomnieć.
Aha, jutro wyjeżdżamy, zbiórka o 8:00 przy auli kryształowej. Wyjeżdżamy do Rogowa. Jeżeli jeszcze ktoś ma ochotę dołączyć, to bardzo gorąco zapraszam i bardzo proszę o punktualność, bo autokar nie może czekać, bowiem w Rogowie mamy wiele atrakcji przygotowanych.
To może tutaj. A część może. Część może
Ja mogę dobra. To by było lepiej, nawet ktoś mógłby potem na przykład klęknąć na stole i byłby wyższy. Pani dyrektor,
Ale proszę. Chodźcie, chodźcie.
Jest takie.
Olka, co ty sobie robisz? Super, dziękujemy.
To jeszcze parę słów o sprawach organizacyjnych.